Hey, so now we get to talk about the DNA double helix. So an anatomic model of a DNA molecule shows the double helix as a characteristic shape of the DNA molecule. So we're going to be able to learn to describe the double helix of DNA and the process of DNA replication. So DNA base pairs is a big part of the DNA double helix and just DNA in general. So during the 1940s, biologists determined that the bases in DNA had a specific relationship. The amount of adenine was equal to the amount of thymine. The amount of guanine was equal to the amount of cytosine. So scientists subsequently determined that adenine is always going to be paired with the thymine, where guanine is always going to be paired with cytosine. So that means that the number of purine bases, adenine and guanine, has to equal the number of pyrimidine bases, or thymine and cytosine. So remember, in DNA, A always binds to T, C always binds to G. Okay? So in comes Watson and Crick. In 1953, James Watson and Francis Crick proposed that DNA was a double helix that consisted of two polynucleotide strands winding about each other like a spiral staircase. This contained a sugar phosphate backbones that was the same to the outside. It was basically like analogous, so the same as describing outside stair railings with the bases arranged like steps along the inside. And one strand goes from 5' to 3' direction, and the second strand is going to go in the anti-parallel direction, or the 3' to 5' direction. So DNA contains complementary base pairs. Complementary is just a fancy word for matching. Okay, if I'm putting together a complementary outfit, I know that red and black go together, but red and pink do not. So complementary base pairs with equal amounts of A and T and equal amounts of G and C bases. So that means that adenine is linked only to thymine, and this happens through the course of 2 hydrogen bonds, and guanine is linked only to cytosine, and this is by 3 hydrogen bonds. So you can see here, A is bonded to T through 2 hydrogen bonds, where G is bonded to C by 3 hydrogen bonds. So a double helix has 2 strands of nucleotides that are going to wind together, and it's held in place by 2 hydrogen bonds that form between A and T, or 3 hydrogen bonds that form between the base pairs G and C. So the 2 strands of the double helix are held together by those hydrogen bonds, again, that are linking the A and T, or to the C to the G. So write the complementary base pair sequence to the matching strand in the DNA section here. Pause me and come back. So A binds with T, G to C, T to A, C to G, C to G, A to T, A to T, T to A, and C to G. So great job, you guys.